Hello dear students, today we will discuss about the topic that is organization of cell nucleus and nuclear bodies and its significance, chromosome movements during cell division. First we will look at the main objectives of today's topic, description and structure of nucleus, nucleoplasm and its components, nuclear bodies and their significance, chromatin organization in nucleus, organization of nucleolus, significance of nucleus, chromatin movements during cell division. Now we will discuss the first part of the topic, structure and organization of nucleus. Nucleus is a membrane-bound organelle found in eukaryotic cells that contains the cell's hereditary information and controls the cell growth and reproduction. It is the most prominent organelle of the cell. The number of nuclei in a cell varies. They may be uninucleate, binucleate, or even multinucleate. The shape of nucleus is mostly round. It may be oval, disc-shaped, depending on the type of cell. Nucleus was the first cell organelle to be discovered. Antony van Leeuwenhoek observed nucleus in the red blood cells of salmon. Robert Brown in 1833 named and discovered nucleus in plant cells. The word nucleus is derived from a Latin word nucleus, which means kernel or seed. The nucleus is the controlling center of the eukaryotic cell. It contains the genetic material of cell. Most eukaryotic cells have one nucleus, uninucleate, but some have many nuclei, multinucleate, such as skeletal muscle cells, and certain cells such as mature red blood cells do not have nucleus. The multinucleate condition of a cell is called syncytium or coinocyte. Some protozoans such as paramecium have two kinds of nuclei, a macronucleus and a micronucleus. Genes in the macronucleus control the everyday functions of the cell, such as feeding, waste removal, and maintenance of water balance. But micronucleus controls the sexual reproduction. Nuclei differ in shape depending on the cell type. Most nuclei are spherical, but multilobed nuclei are also common, such as polymorphonuclear leukocytes. A nucleus has following components: nuclear envelope, nucleolus, nucleoplasm, and chromosomes. Nucleus accounts for about 10% of the cell's volume. Nucleus is separated from the rest of the cytoplasm by a nuclear membrane. Now, structure of cell nucleus. The cell nucleus consists of a nuclear membrane, nuclear envelope, nucleoplasm, nucleolus, and chromosomes. Nucleoplasm, also known as karyoplasm, is the matrix present inside the nucleus. Let us discuss in brief about the several parts of a cell nucleus. The nucleus is considered to be one of the most important structure of eukaryotic cell, as it serves the function of genetic information storage and duplication of genetic information. It is a double membrane-bound organelle that harbors the genetic material in the form of chromatin. Nucleus consists of a dynamic mixture of non-membranous subcompartments of various functional capacity. The key functions of the cell nucleus include deoxyribonucleic acid replication and further to control gene expression during the cell cycle, transcription and further post-transcriptional processing of pre-messenger ribonucleic acids occur inside the nucleus, and the mature mRNAs are transported into the cytoplasm where the translational events occur. Thus, nucleus provides functional compartmentalization inside the cell, allowing higher level of gene regulation. Now, nuclear envelope. The nuclear envelope is a double-layered membrane that encloses the contents of the nucleus during most of the cell's life cycle. The space between the layers is called the perinuclear space, and this space is continuous with the rough endoplasmic reticulum. The envelope is perforated with tiny holes called nuclear pores. These pores regulate the passage of molecules between the nucleus and the cytoplasm, permitting some to pass through the membrane but not others. The inner surface has a protein lining called the nuclear lamina, which binds to chromatin and other nuclear components. During mitosis or cell division, the nuclear envelope disintegrates but reforms as the two cells complete their formation and the chromatin begins to disperse. Now, nuclear pores. The nuclear envelope is perforated with holes called nuclear pores. The average mammalian cell contains several thousand nuclear pores. Each nuclear pore is formed from an elaborate structure called nuclear pore complex. The proteins that make up the nuclear pore complex are known as nucleoporins. These pores regulate the passage of molecules between the nucleus and cytoplasm, permitting some to pass through the membrane but not others. Building blocks for forming DNA and RNA are allowed into the nucleus, as well as molecules that provide the energy for constructing genetic material. Now, nuclear skeleton. Just beneath the nuclear membrane is a meshwork of thick filaments called nuclear lamina, which are intermediate filaments. The nuclear lamina provides mechanical support to the nuclear envelope and serves as a site of attachment for chromatin fibers at the nuclear periphery. The lamina is composed of polypeptides called lamins. Mutation in lamina gene causes a disease called Hutchinson's-Gilford progeria syndrome that is characterized by premature aging and death. Now we will move to next part of the topic, nuclear bodies. The nucleus is separated from the cytoplasm by a double membrane. The outer nuclear membrane is continuous with the endoplasmic reticulum. The nucleus is compartmentalized and contains numerous subnuclear structures called nuclear bodies. The nuclear bodies present in nucleus are nucleoli, splicing speckles, cagal bodies, gyms, and promyelocytic leukemia (PML) nuclear bodies. In contrast to cytoplasmic compartments, the subnuclear bodies lack a membrane separating them from the nucleoplasm. These subnuclear bodies may serve to enhance the efficiency of specific nuclear processes. Now, nuclear speckles. Nuclear speckles, also known as interchromatin granule clusters, are irregular shaped structures of varied size, and the nucleus typically contains 25 to 50 of these subnuclear bodies. Nuclear speckles are enriched in pre-mRNA splicing factors, including small nuclear ribonucleoprotein particles and non-SNRNP protein splicing factors. Speckles are often found close to actively transcribed genes and are thought to act as a reservoir for the splicing of nascent pre-mRNA at nearby genes. Now, cagal bodies. Cagal bodies are roughly spherical structures numbering 1 to 5 per nucleus, varying in number and size. These structures appear in the form of coiled threads and are characterized by the presence of the coilin protein. The cagal bodies are not seen in all cells or tissues, but are especially prominent in highly proliferative cells such as cancer cells or metabolically active cells. Cagal bodies are thought to play a role in SNRNP biogenesis and in the trafficking of SNRNP and small nucleolar RNPs. Cagal bodies are rich in spliceosomal U1, U2, U4, U6, and U5 SNRNPs, as well as U7 SNRNP involved in histone 3' end processing. Most histone transcripts are not polyadenylated, rather their 3' ends are produced by an endonucleotic cleavage, and U3 and U8 SNORNPs involved in processing of pre-rRNA. Now, promyelocytic leukemia (PML) nuclear bodies. PML bodies are characterized by the presence of PML protein. PML bodies vary in size from 0.3 to 1 micron meter in diameter, and a nucleus typically contains 10 to 30 of these structures. The primary role of PML bodies remain unclear, but they may play a role in transcriptional regulation and in antiviral response. PML nuclear bodies have emerged as important regulators of cell cycle, defense against viral infection, induction of apoptosis, and cellular senescence. PML proteins may act as a tumor suppressor. Cellular stress such as inflammation and oncogenic stimulation cause an increase in the number and size of PML nuclear bodies. Now, significance of nuclear bodies. Nuclear bodies help in splicing, that is removal of non-coding introns. Some nuclear bodies, for example cagal bodies, contain some molecules called small nuclear RNPs and small nucleolar RNPs, which help us in the splicing of some non-coding sequences from mRNA and rRNA. Nuclear bodies help us in organization and formation of ribosomal subunits. Some nuclear bodies act as transcriptional coactivators and thus enhance gene expression. Nuclear bodies such as PML nuclear bodies act as tumor suppressor protein, provide defense against viral infection and induce apoptosis. Now, nucleoplasm. Nucleoplasm or karyoplasm is the protoplasm within the nucleus. It is enveloped by the nuclear membrane. The nucleoplasm includes the chromosomes and nucleoli. Many substances such as nucleotides necessary for purposes such as DNA replication and enzymes which direct activities that take place in the nucleus are dissolved in the nucleoplasm. The soluble liquid portion of the nucleoplasm is called the nucleosol or nuclear hyaloplasm. The main function of the nucleoplasm is to serve as a suspension substance for the organelles inside the nucleus. It also helps us maintain the shape and structure of nucleus and plays an important role in the transportation of materials that are vital to cell metabolism and function. Now, chromosomes. Chromosomes are present in the form of strings of DNA and histones called chromatin. A chromatin is an organized structure of DNA and proteins that is found in the nucleus of eukaryotic cell. Chromatin and chromosomes are basically the same thing. The difference is that chromatin is less condensed and extended DNA, while chromosomes are highly condensed DNA. The word chromosome comes from the Greek word chroma, color, and soma, body, due to their property of being very strongly stained by particular dyes. Chromatin is further classified into heterochromatin and euchromatin based on the functions. The heterochromatin is highly condensed, darkly stained and transcriptionally inactive form, mostly present adjacent to the nuclear membrane. On the other hand, euchromatin is less condensed portion of chromatin. This region is transcriptionally active and contains most of the transcribing genes. Organization of chromatin, which is found abundantly in a transcribing cell. The basic proteins in chromatin called histones are rich in basic amino acids, lysine and arginine. There are 5 types of histones found in chromatin: H1, H2A, H2B, H3 and H4. The DNA and histones are organized into repeating subunits called nucleosome. Each nucleosome contains a nucleosome core particle consisting of 146 base pairs of DNA wrapped around a disc-shaped complex of 8 histone molecules. The histone core of each nucleosome consists of 2 copies each of histone H2A, H2B, H3, H4, assembled into an octamer. The remaining histone H1 resides outside the nucleosome core particle. The histone H1 is called linker histone because it connects one nucleosome core particle to the next. Now, nucleolus and its organization. The nucleolus is a non-membrane nuclear body which disappears in the late prophase and reappears in the telophase stage of cell division. It is observed in interphase cells that are actively synthesizing proteins. More than one nucleolus can be present in the nucleus. Each nucleolus is produced by a nucleolar organizing region of a chromosome, which is termed as nucleolar organizing chromosome. It contains mostly rRNA and proteins along with a modest amount of DNA. It possesses nucleolar organizer region portion of the chromosomes. In humans, chromosomes 13, 14, 15, 21 and 22, where rRNA genes are located. These regions are involved in reconstituting the nucleolus during the G1 phase of the cell cycle. The nucleolus contains 4 distinct regions. The nucleolus is a dense spherical shaped structure present inside the nucleus. Some of the eukaryotic organisms have nucleus that contains up to 4 nucleoli. The number of nucleoli is different from species to species, but within a species the number is fixed. The nucleolus plays an indirect role in protein synthesis by producing ribosomes. These ribosomes are cell organelles made up of RNA and proteins. They are transported to the cytoplasm, which are then attached to the endoplasmic reticulum. Ribosomes are the protein-producing organelles of a cell. Nucleus disappears when a cell undergoes division and is reformed after the completion of cell division. Nucleus is the site of transcription of ribosomal RNA and assembly of ribosomes. It is responsible for synthesizing a large nascent precursor ribosomal RNA, that is 45S in mammals, and the processing of this RNA to yield mature ribosomal RNA of 18S, 5.8S and 28S. The assembly of these RNAs with incoming ribosomal proteins generates small and large ribosomal subunits that then pass into the cytoplasm. Now, functions of cell nucleus. The cell nucleus acts like the brain of the cell. Speaking about the functions of the cell nucleus, it controls the hereditary characteristics of an organism. This organelle is also responsible for the protein synthesis, cell division, growth and differentiation. Here is a list of the important functions carried out by a cell nucleus. It controls the hereditary characteristic of an organism. Nucleus is responsible for protein synthesis, cell division, growth and differentiation. Store proteins and RNA in the nucleolus. Stores hereditary material in the form of deoxyribonucleic acid strands. Nucleus is a site for transcription in which messenger RNA are produced for protein synthesis. Exchange of hereditary molecules between the nucleus and the rest of the cell. During the cell division, chromatins are arranged into chromosomes in the nucleus. Production of ribosomes in the nucleolus. Selective transportation of regulatory factors and energy molecules through nuclear pores. Nucleus helps in the processing of mRNA or RNA and make these molecules functional. It regulates the integrity of genes which regulate the gene expression, in turn regulating the activities of cell. Therefore, the nucleus is known as the control center of the cell. Aids in exchange of DNA and RNA hereditary material between the nucleus and the rest of the cell. Now, chromatin movements during cell division. To maintain continuity of life, the cell must replicate its DNA in S phase of cell cycle, and the identical copies of DNA are equally distributed to its daughter cells. This process of replication and division of cells make the chromosome highly flexible and dynamic structure, and accordingly they change their morphology and position. The most dramatic chromosome movements occur during mitotic and meiotic divisions, when daughter chromosomes or homologous chromosomes are distributed to daughter cells at the onset of anaphase. The long distance movements of condensed chromosomes during mitotic and meiotic cell divisions are mediated by spindle microtubules inserted at the kinetochore during cell division. Now we will discuss the division of cell and accordingly the chromosome movements during cell division. Cell cycle. The cell cycle is a sequence of events or changes that occur between the formation of cell and its division into daughter cells. It has a non-dividing growing phase called the interphase and a dividing phase called a mitotic or M phase. The most important part in cell which plays a role in efficient segregation of duplicated chromosomes is centromere. The centromere radiates many types of microtubule fibers which gets attached with the kinetochore of duplicated chromosomes and pulls them to opposite poles of cell at anaphase. The radiating microtubule fibers and their association with chromosomes at equator of cell forms a structure called spindle apparatus. The chromosome movements during cell division is chiefly due to the spindle apparatus. This apparatus contains three different types of microtubules. Astral microtubules radiating from centrioles found in animal cells only, continuous or interpolar microtubules passing from one pole to the other accounts for the bulk of spindle structure, and chromosomal or kinetochore microtubules which originate from the poles and are attached to the centromere. Now, interphase. It is a long metabolically active phase between two successive mitotic cell divisions. It has three substages. G1 phase. During this phase, growth of cell occurs and prepares for DNA, RNA, and protein synthesis. S phase. Synthetic phase. During this phase, duplication or replication of DNA and centrioles take place. G2 phase. During this, synthesis of proteins required for the synthesis of spindle fibers takes place. Now, M phase. It is a short phase. It includes two important processes that occur simultaneously. They are karyokinesis and cytokinesis, resulting in two daughter cells. After M phase, the cell may enter either interphase to repeat the cell cycle or G0 phase to arrest cell cycle. Then the cells in G0 phase may grow and differentiate into different cell types to perform different functions. Mitosis. Walter Fleming studied mitotic cell divisions in animal cells and coined the term mitosis. Mitosis is a type of cell division in which a parental cell produces two similar daughter cells that resemble the parent cell in terms of chromosomal number. So, it is also called equational cell division. This maintains constant number of chromosomes in each cells of successive generation. It occurs in somatic cells of the body, so it is also called somatic cell division. Mitosis occur in two stages, that is karyokinesis and cytokinesis. Now, karyokinesis. It is a division of nuclear material. It occurs in four stages as follows. Prophase. It is the longest phase. During this phase, the chromatin is organized into distinct chromosomes by coiling or spiralization. The centrioles develop into esters and move towards the opposite poles of the cell to establish the plane of cell division. Spindle apparatus begins to appear. Nucleus and nuclear membrane disintegrates and disappears. The chromosomes are set free in the cytoplasm. Esters are not formed in plant cells as they do not have centrioles, hence called anestral cell division. Now, metaphase. Spindle fibers are completely formed. The chromosomes become short and thick with two distinct chromatids each. All the chromosomes move towards the center of the cell and arrange in the equatorial plane, right angles to the position of esters to form metaphasic palette. Chromosomes are attached to spindle fibers at their centromere. Now, anaphase. The centromere of all the chromosomes undergo longitudinal splitting and the chromatids of each chromosome separate to form daughter chromosomes. The daughter chromosomes move towards the opposite poles from the equator by the activity of spindle fibers. Now, telophase. During this phase, the events of prophase will be reversed. The daughter chromosomes reach the opposite poles. The chromosomes undergo decondensation to form long, thin, thread-like structures called chromatin. Nucleus and nuclear membrane reappears. The spindle fibers disappear. Now, cytokinesis. Cytokinesis is that part of cell division process during which the cytoplasm of the cell divides into two daughter cells. Cytokinesis starts during the anaphase and continues through telophase. A ring of actin filaments called the contractile ring forms around the equator of the cell just beneath the plasma membrane. The contractile ring shrinks at the equator of the cell, pinching the plasma membrane inward and forming what is called a cleavage furrow. The cleavage furrow develops in the middle of the cell in centripetal direction due to the contraction of actin filaments. The cleavage furrow contraction continues till the edges of the plasma membrane meet and form two complete cells. In plant cells, the cytokinesis cannot proceed with a cleavage furrow due to their cell walls. Instead, a cell plate forms across the cell in the location of the old metaphase palate. Some membrane enclosed vesicles derived from the Golgi apparatus migrate to the center of the cell where the metaphase palate used to be and fuse to form a cell palate. Eventually, the growing cell palate fuses with the existing plasma membrane, producing two daughter cells, each with its own plasma membrane. With this, we end today's topic. Thank you.